Greetings to you all. This is teacher Dr. Mgombo Ronald. In this video, we are going to go ahead and look at the process of the Calvin cycle. That's what we are looking at today, the Calvin cycle. Okay. Then we are going to go ahead and look at the Calvin cycle. So previously, we looked at what we called the light-dependent reactions. So today we are going to look at the light-independent reactions, or the Calvin cycle. They call it the light-independent reaction simply because it does not require light for it to take place. That is why they call it the light-independent reaction. It all begins with carbon dioxide being received by ribulose biphosphate in the presence of an enzyme known as, so usually it all begins with an enzyme, the receptor known as ribulose bisphosphate. This receptor here known as ribulose bisphosphate is usually in the presence of an enzyme known as ribulose bisphosphate carboxylase. This receptor here is a 5-carbon molecule. So after taking up carbon dioxide, it forms what we call a 6-carbon intermediate. But this 6-carbon intermediate is very unstable. So since it is very unstable, it splits immediately into 2 molecules of glycerate 3-phosphate. Since we have now 2 molecules, so we shall have 2 molecules of GP, or glycerate phosphate. Okay, we have 2 molecules of glycerate phosphate, or glycerate 3-phosphate. That whole stage is known as the carboxylation stage of the Calvin cycle. The next stage we are going to see is the stage that is known as the reduction stage. During the reduction stage, that is where we use the products from the process of the light-dependent reactions into the light-independent reactions. From the light-dependent reactions, we had reduced NADP and we also had ATP. Those were the products that we formed from the light-dependent stage of photosynthesis in the thylakoids. All these reactions, known as the Calvin cycle, takes place in the stroma of the chloroplast. Now, what usually happens is we have what we usually call this reduced NAD is usually converted into oxidized form. The reduced NAD is converted into an oxidized form that donates protons, and these protons are received by glycerate 3-phosphate, and that is very important for converting glycerate 3-phosphate to a triose phosphate. So we end up with a triose phosphate. This word here is triose phosphate. Okay. Then another molecule that is usually converted or broken down is ATP. ATP is converted to ADP and an inorganic phosphate. That enables us to form triose phosphate. So what happens is now this triose phosphate is going now to go ahead and be converted. It is dimerized through a process known as dimerization. What usually happens, and in this reaction here that we are seeing that converts glycerate 3-phosphate to triose phosphate, also we see some emission of water molecules. So we usually see some water being produced from this reaction. After that is done, we have the triose phosphate. Now this triose phosphate here is the one that is usually dimerized to form glucose. So it undergoes a number of reactions. It can be converted, it can be directly used to produce energy in respiration, but it can also be dimerized to form glucose. Okay, so it dimerizes and forms glucose. Now, this glucose molecule can be used for a number of ways. So it can form the glucose molecule. The glucose molecule can undergo polymerization to form starch. The glucose molecule can be converted into lipids. The glucose molecule can also be converted into amino acids in the plant body. But besides that, it is believed that most of the triose phosphate is used to regenerate the carbon dioxide acceptor known as ribulose biphosphate. So, most of the triose phosphate is not converted to glucose. Most of the triose phosphate is usually converted to ribulose biphosphate, which is now going to go ahead and combine again with carbon dioxide, and that reaction and the whole reaction continues. So this whole stage where we see formation of ribulose biphosphate from triose phosphate, that is what they call the regeneration stage. So what we have done today, we have looked at the Calvin cycle, all the dark reactions. It happens within the stroma of the chloroplasts under the influence of an enzyme known as ribulose biphosphate. This specifically happens in C3 plants. Why are they called C3 plants? Because their first carbon intermediate has 3 carbons. It all begins with carbon dioxide combining with ribulose biphosphate. When that happens, in the influence of ribulose biphosphate carboxylase enzyme, it releases a 6 carbon intermediate. This 6 carbon intermediate, being very unstable, it splits into 2 molecules of glycerate 3 phosphate. When it does so, glycerate 3 phosphate is usually undergoing reduction, where we see the products from the light dependent reactions, that is to say, reduced NADP and ATP being used. In the due course, we shall see reduced NADP being converted to oxidized NAD, and then ATP being converted to ADP and an inorganic phosphate that produces a molecule known as the triose phosphate. This triose phosphate is central for synthesis of very many different molecules. First, the triose phosphate can be converted to glucose. 2, the triose phosphate can also be converted to ribulose biphosphate to regenerate the carbon dioxide acceptor. The glucose that is formed can either be polymerized to form starch, or it can be converted to amino acids, or it can be converted to lipids, different molecules which are synthesized in the plant body. I remain teacher Dr. Mugomba Ronald. Consider to subscribe. Consider to share, consider to like, and drop in your comments. I remain teacher Dr. Mugomba Ronald. Till next time.